All right. Good afternoon, everyone. So I am a synthetic biologist. I think about using math to represent things and doing theory and computation. But since I'm a driver talk, I wanted to kind of talk a little bit about what my group does and maybe try to inspire this group to think about different kinds of problems that go beyond protein-DNA interactions and protein evolution. Not that those things aren't amazing and I don't love them, but just to kind of think, well, bigger, better, more complicated, and how can we get there? And to give you an idea about where my background comes from, so I started actually as a protein engineer with Francis Arnold, thinking about how we engineer proteins involved in quorum sensing and gene regulation. And I love that those types of projects because you're using a forward engineering approach, right? Making something do something different in order to understand how it works. And so now I try to apply that thinking about organisms in a community type of setting. And so why am I obsessed with microbial communities? I love them as an engineer because you have a division of labor amongst organisms. And so if I think about this from a building something, making something new perspective, this allows me to reduce metabolic load on each individual organism in a community. So instead of asking organism to do a little bit more and a little bit more and a little bit more and it losing fitness in each of those steps, if I divide it amongst more organisms, can I have higher fitness overall and make the system work really well? Can I take advantage of different capabilities of organisms? So instead of just using my favorite E. coli and manipulating it to do what I want, that actually doesn't work a lot of the times. There are lots of enzymes that don't function in E. coli that we'd like. Can we take advantage of the capabilities of other organisms? And I'll talk a little bit about that. And then modularity, right? I think in information processing and other types of things, we talk about different modules, how they can be plug and played and swapped. So instead of thinking about those at a codon level, can you think about those at an organism level? And what does that allow you to do? And what are the constraints on these systems? And so, I think about this as an engineer. So what are the kind of applications we think about? So biofuel production. Can you distribute breaking down cellulosic biomass with making something useful? Maybe fuels aren't the best economic choice, but we'll see. Can you make different types of biopolymers like plastics? Can we develop systems that will allow us to extract different types of minerals, et cetera, from mining samples? Can we replace traditional fertilizers with mixtures of microbes that will allow organisms to resist different types of changes in the environment and get different types of nutrients instead of having to add chemicals that may have other kinds of effects? And then, of course, the human microbiome. I do think about kind of top-down approaches to understanding microbiomes and how they might change and adapt. I've kind of taken that out of the talk today just because of the interest of time. And so as with my synthetic bio hat on, right, I try to think about what do I need to understand and what are the different components that I need to think about in order to make a community that functions the way I want, makes something useful, or carries out some sort of complex task. And so to do that, I need a few different things. And so we can think about how do I manipulate community composition? How do I get the organisms to grow together when I want them to do with the dynamics or the stability that I'd like? How do I get them to coordinate their behavior? How do I get them to talk to each other so that they do what I want when I want them to, or they have feedback mechanisms amongst each other? And then how do I think about dividing up functions amongst those community members? And so since communication is one of the elements of this, this area, I wanted to talk a little bit about what we can do with cell communication. And so my group has been interested in using a model co-culture system made up of E. coli and bacillus. And so for those of you a little further away, right, E. coli is a gram-negative organism. It's a workhorse in terms of biological engineering, protein engineering, et cetera. Bacillus megaterium is a gram-positive organism, and so it's kind of on this other divide, evolutionary divide of types of microorganisms. It has a different type of membrane structure, et cetera. This other organism, can maintain plasmids. It's used for industry. It has a lot of useful things for other applications like protein secretion mechanisms. And so the interesting thing is that there are these types of cell communication systems. And sometimes these types of organisms can talk to each other using a single communication system that Bill Bentley is an expert in, which is AI2. But we wanted to develop some other communication tools that allow these organisms to talk to each other. And so can we rewire existing intraspecies communication systems to enable interspecies communication? And so I want to get these things to talk to each other. And so to do this, we can use acyl homoserine lactone dependent expression. And so in these types of systems, you essentially have two different behaviors. So you have at low cell density, we have no gene expression. The cells produce this small molecule and it accumulates as you get to higher cell density. This molecule is recognized and you turn on gene expression. My group started working with an analogous protein, which is ESA-R. Excuse me. And essentially, in this case, ESA-R blocks transcription here. You add the same molecule and you turn on gene expression. And so the details, right, I don't want to get lost on, is you have the same logic. So add this little red molecule, this quorum sensing molecule, you turn on gene expression. But the logic and the biochemistry behind it are a little bit different. The really nifty thing from an engineering perspective, I think, is that if you swap the location of this ESA binding box and this promoter, you can actually turn this system into an activation system, where in the absence of signal molecule, you turn on gene expression, you add the signal molecule, the protein leaves the DNA, and then you get gene expression turning off, right? And so this is a typical kind of a biological, beautiful system, right? You turn some genes on and some genes off in response to the same signal molecule. But from a biotech and bioengineering perspective, we don't have a lot of these tools at our disposal. And so we can do things like engineer these systems. So we can use directed evolution and make a library of these proteins, and then have ones that operate over about 4 orders of magnitude that we can then swap and plug and play with as we're engineering different types of systems. All right, so far I've shown you that E. coli can recognize this acyl homoserine lactone signal. So we can also engineer bacillus to recognize or to produce the signal. So as I mentioned, right, if you think about these grand types of quorum sensing systems, the gram negatives like E. coli, right, it's other types of gram negatives produce and respond to these acyl homoserine lactones, but these signals have never been seen in gram positive organisms. And so we were hopeful that the enzymes would still work. And so we expressed it and in a xylose-inducible manner, isolated supermate, and then used an E. coli reporter system to see if we could get, essentially in this case, cherry or a luminescent signal. Our initial efforts didn't work, but we were able to essentially tune the conditions in a way that enabled it to work. I won't tell you about those in detail, but you can kind of see, right, and depending on what kind of resolution you like to see, you can see that if I culture these bacillus, which are kind of white here, with our E. coli, which have now turned pink because they're recognizing signal. Now we have an artificial cell-cell communication between these two organisms that don't naturally communicate with each other, and in all likelihood probably wouldn't interact with each other very often in the real world. And we can see this quantitatively here, where we titrate the starting concentrations of the two organisms and see that we do see nice cell-cell communication of them when they're coexisting. Okay. We can then take another type of communication system, one that doesn't exist in bacillus or in E. coli, and we can take the signal response capabilities, which are encoded by a 2-component AGRC and A system, as well as a promoter, and we've just put a GFP reporter here. And then we have the signal production components, which we're going to put in E. coli. And so this is a pre-peptide, this AGRD, and a protease. And essentially it takes this AGRD peptide, processes it, and you end up, in this case, with a small molecule signal here that is a peptide. So it's an 8-mer with a thiolactone ring. And when it accumulates, it changes the gene expression through recognition of this AGRC and down and back. And so without going into too much detail, if I put these components in the E. coli to produce a signal and the bacillus to recognize it, and I titrate the relative amounts of the two organisms, I can see good communication, and this indicated by fluorescence, through the communication using this acyl homoserine, or this AIP molecule. And so this gives us a platform for two-way communication, right? And this is, right, alongside any natural communication between these organisms that might happen from sharing nutrients or other types of things. And so this allows us some new tools to coordinate behavior of very different types of organisms in synthetic communities. No, wait. All right, so moving on, we can think about how do we alter community composition. So from a biochemical engineering perspective, right, you look at the very early iterations of biochemical engineering, and Bailey, Schuler, et cetera, were like, okay, let's grab all the microbes that have all the different functions we want, and let's put them together, and let's build the functions, right, the community that has the capabilities that we're looking for. And then all along came, right, the recombinant DNA revolution, and since then, most of, right, the research is focused on let's take all those different functions and put them in a single microbe and grow them in a single system. And granted, for a lot of things that might be more, right, might be simpler. You don't have to worry about co-culture balance of different organisms, but there's different capabilities that we can combine and use mixtures together. But if we're going to do that, we need a new set of tools to do that. And maybe through that, we can learn about natural ecological interactions as well. And so my group is focused on synthetic or on metabolic complementation or cross-feeding in order to enable this type of mutualistic interaction. And so our work is built on work from other synthetic biologists and ecologists that has shown that in a little bit more frequently than one might have anticipated, if you take a pair of auxotrophs, so two organisms that are not able to grow on their own because they're missing one essential gene. And of course, that's relative to the environment they're in. So we're talking about pairs of amino acid auxotrophs. And so those pairs, each of them is unable to make one essential, right, amino acid. Right? So for us, we have to get a bunch of essential amino acids from our diet. Right? A lot of E. coli and other organisms can make all the amino acids. So I can knock out one of the pathways and make them require that amino acid from the environment. And so if I take an amino acid auxotroph and put it with another one, I can essentially see that they can get growth together. And so here's a quick illustration of that. So I have a leucine auxotroph for Bacillus megaterium, and E. coli auxotrophs from the KO collection. In this case, tyrosine, isoleucine, and arginine. And at the top, I've essentially spot plated E. coli on its own. On the top, it's Bacillus megaterium. And on their own, in this type of media condition, the cells don't get the amino acids they need, and essentially they die. Whereas if I grow them together, right, they'll actually cross-feed and you see growth. And so I can look at this for a set of pairs. So I have my leucine auxotroph of Bacillus, and then I look at all of the amino acid auxotrophs from the KO collection, which is a collection of gene knockouts. And so I can see, right, for a lot of these, right, so I don't see any growth on Bacillus or E. coli on their own, except for the one case of this phenylalanine here. But I do see good co-culture growth, right, for isoleucine, arginine, methionine, and proline. And so this cross-feeding is working, where essentially under some of these conditions, these two organisms are getting the required amino acid from their partner. We can then do the same thing in batch culture. In this case, for each of these plots, we're looking at E. coli growth on its own, E. coli growth with Bacillus. So in this case, this isoleucine, it grows very nicely. Bacillus co-culture growth, and then Bacillus monoculture growth. And so with this setup here, you can see that we see really nice co-culture growth, whereas the monocultures don't grow on their own. We have some where we see a little bit of co-culture growth, or the co-culture stays alive, and then a set here where there's no interactions between the two organisms. And then, of course, right, as biologists and engineers, we'd like to look at this in continuous culture. There's lots of opportunities for bioengineering in continuous culture, and also it adds the potential to ask evolutionary questions as well. So we built a continuous culture system. We have about 20 mil working volume in these. We're aerating and mixing using aquarium pumps, and then using a heat block to maintain temperature. And so over time, we can see that we actually do see co-culture growth. And so in this case, we have a period of time where we're keeping the volume in our reactors the same and allowing the cells to adjust to their environment. And then we turn on the pump, and we can see that our monocultures, which are these dotted lines of E. coli and Bacillus, they wash out rather quickly. but the co-cultures essentially grow and maintain a steady state concentration together. And so they are complementing each other, and they're getting those amino acids from each other. And so this works in 3 of our co-cultures. Arginine didn't cross-feed as well, and it essentially washes out after a little bit of time. This phenylalanine one, which looked like it had a little bit of growth on its own, right? The cross-feeding is not sufficient in order to support co-culture growth. Right? And so this is very observational. All I've shown you is growth so far. Right? So how do we move to something a little bit more sophisticated? And so hopefully we can move simple models of ecological interactions can inform our understanding of complex systems. So can we go from gene circuits to systems biology? Can this go to co-cultures and microbiomes? So can we use these types of model systems to ask questions? And so what I didn't tell you is we've also been doing RNA-seq analysis for these communities, and we can see that even though we're seeing cross-feeding, the way that the cells are responding to being grown together is not the same for all of the different organisms. And we're also looking at the amino acids that are being secreted. And so, are the same amino acids being secreted in all of these conditions? Are these organisms just like, hey, you put me together, I'm growing, I don't really care, I'm not really doing anything different, I just get this thing from the environment? Or are they responding in a more wholesale way that's changing how they adapt and what their long-term trajectories might be? And so just as Steve was talking, are there rules for amino acid substitution and protein engineering rules, Are those rules going to be similar to rules for substituting organisms in dynamic communities? How do we think about pathway? Is there a pathway effect? Is it more like a state function? We don't really know. How do we start to ask those questions in a really systematic way of more complex systems and ecological systems? Okay. And I think one of the challenges for us is also how do we integrate a range of data types, and how do we do this in a real-time fashion? I know people thinking about microbiomes think about this frequently, but I think that there's a cluster of information in this room that might be valuable to help us think about these questions as well. Okay. So I want to talk a little bit in the last few minutes about reaction and pathway engineering. We've thought about this in a range of different ways. Some of which are distributing functions amongst different organisms so that we have our bacillus secreting a protein that allows us to have cellulose degradation. So cellulase is enabling cellulose degradation. And then E. coli taking up those sugars that are released to make some sort of pharmaceutical molecule or biofuel. And the other types of applications we're thinking about are biosensing. And so I thought our biosensing application might be a little bit more relevant to this kind of conversation. And so, for biosensing, we can think about inputs of small molecules or metals coming in, some sort of logic circuit, information processing, and then some sort of actuation. And so in my group, of course, we'd like to divide sensing and actuation roles amongst different organisms. That allows us to take advantage of that modularity and perhaps have even more complicated computation. And you can also, of course, do things like signal amplification, et cetera. So in my group, we've done things like engineer AND gates, which integrate quorum sensing signaling, so density-dependent recognition using quorum sensing plus some sort of external signal. To do this, we've built an AND gate where you only have gene expression turning on when you have both the quorum sensing signal plus an external signal, in this case, an hydrotetracycline. And we can see as the cells grow, only when you have all of the components and high cell density, essentially information got removed here, but do you see gene expression turning on? And then other groups have taken a similar concept, and you can actually put AND gates in multiple organisms in a row in order to detect combinations of different molecules. And only when everything is present do you see that kind of output you're looking for. So in my group, we were looking at motility as a biosensing output. And this is the input in this case is the quorum sensing molecule. And so the way we built this, essentially, we're using a MOT-A knockout. And so MOT-A is a key motor protein involved in cell motility. And so they can make flagella, they just can't spin them. And And so when we take this gene knockout here, we have no motility, and that's analogous to this illustration here where we have a petri dish and we have a low agar concentration media, and essentially we stab the cells in the middle and they can't move through the media. If we add back this MOT-A protein under the control of the ISA-R protein, ISA-R promoter, which is constitutive in the absence of ISA-R, we see that our swim radius increases, which is this illustration here. If we add in ISA-R, which is the repressor, we actually turn off gene expression again. And then if we add the acetylmyoserine lactone, right, to turn on transcription once again, by inducing ISA-R, we see, once again, we can see swimming. So the system works as we expect. We found that if we made a gradient of acetylmyoserine lactone, so in this case, in the 100 nanomolar and 1 micromolar, essentially we added the acetylmyoserine lactone to these Petri or these Whatman filter papers, let them diffuse for a little while, and then stabbed the cells in the middle once again. That not only did we see the radius that we expected, we also saw that the cells were moving up the gradient. And this was really interesting to us because, one, we're not manipulating chemotaxis. It's just a yes/no, can they move, right, behavior. And so we thought that they would just move uniformly. And we were actually able to capture that using a simple physical model that incorporates, right, movement of the cells as well as gene expression changes and diffusion of the acetylmyoserine lactone. And using a system where essentially we capture it using an enhanced diffusivity, we're able to incorporate all of these aspects. So the cells, changes in gene expression, motility, and their movement up the gradient. Of course, then we want to, right, have distributed sensing where we have a sensor cell type and a motility cell type. And we can do that. We built a simple system using IPTG. And so we can see as we move up in complexity from our control with no signal to no IPTG and increase the concentration of IPTG, we see the cell radius of the cells as they move increase, and then we see that they move up the gradient towards the source of that quorum sensing signal. And then we end, of course, with trying to do something a little bit more practical, where we made a VOC biosensor. So having the ability to recognize volatile organic compounds can be really helpful and useful. This ties into some other applications in our group. And so in this case, we used a xylene sensor from Pseudomonas putida and encoded that. And so you have our sensor cells here, which recognize xylene and produce the quorum sensing signal via this ISA-I protein. And these other ones that essentially have MOT-A dependent torque. And so once again, we see, right, relative to our control, our background has a little bit of expression, and then we see much more motility with the xylene as well. And so we need a little bit more control in here to have a perfect sensor, but it's moving in the correct direction. And so I just wanted to wrap up. And so thinking about how we can maybe combine bioengineering and the right information aspects and communication aspects here. And so, right, elucidating an engineering complex system requires both experiments, computation, and theory. And so how can information theory be applied more to complex systems? Right, how do we get there? And listening to this morning's talk, I'm not sure I know how to do that, but I'm sure combining heads, we can do that. And how can engineered biological systems can be used more to test these theories, right? Synthetic biology and looking at gene circuits has been really helpful for understanding systems biology. Can we think about that from a system or synthetic ecology type of perspective? And so I'll leave with acknowledgments, and I'm happy to take questions.
I think it's very interesting. I was wondering, in several attempts of synthetic biology to produce, let's say, complex behaviors, there's a scalability problem because, let's say, the components that do something, when you start putting them together, they start interacting instead of being isolated as in traditional electronic circuits.
Yeah.
I don't know whether if you've run into that problem or whether you can have some ideas on the...
So I think it depends on the question you're asking. I think a lot of the early ideas in synthetic biology is like, let's emulate an electronic system. And that's not really what biology is for. And so I think the deeper questions are, how do we make biology work in the context of biology, but for some sort of engineered application? And so you need to think about insulation or isolation sometimes, but maybe you can think about other ways to take advantage of it. And that's one of the reasons my group kind of pushed towards synthetic biology on the application side. Because if it just works in our microfluidic device, then is it really useful, and does it really tell us something about the outside world? I don't think we've solved that problem, but I think using approaches like directed evolution or things like that might be more helpful. Yeah.
Great.
Thank you.
We have time for another question in the back while we disconnect your computer.
Yeah.
Hello.
Hi.
Here at the back. So I was wondering, once you have a tool that works for the purpose you designed it for, how stable is it? Because especially in community ecology, things keep evolving, so they for sure might change something that you didn't intend at the beginning.
Yeah. So that's part of the reason we're working towards building ecological interactions between the organisms, so that they will grow together. So you're not just, I'm going to minimize resource competition and they should coexist, but we're going to build the specific interactions. That being said, evolution is real. Someone's going to have a mutation that allows one organism possibly to take over. What kind of checks and balances, do we need in the system to enable stability? We don't know yet. We're working towards looking at those questions. So we have ongoing experiments right now where we're looking at ramping up that pump rate and seeing what happens. Do we still see long-term coexistence? Also, what kind of biomass levels and biomass productivities can we get so that we can maybe think about really using these for bioengineering applications? But those are great questions, and things that now we're starting to have the tools that we can actually test in a system that's simple enough where we don't have like a billion confounding effects. That's a great question.
Great. Thank you very much, Cynthia.